Good morning, everyone. Welcome to the number one lecture, main lecture of this meeting, I would say, nearly. It's a total honor to say hello to Dr. Robin Patel, and I will tell a little bit more about her in a second, but it's our society, ASIC, knows one important prize that was last time around, 3 years around, won by John Turnage, who is here. I had asked him, can I say, you have the smartest winner ever, but actually I'm not allowed to say that. So, but it's a total honor to have selected, and for Robin Patel, who is and she has so many titles, I only mention a few. The Elizabeth P. and Robert Allen Professor for Individualized Medicine, Director of the Infectious Disease Research Laboratory, Co-Director of the Clinical Laboratory, Vice Chair of Education in the Department of Laboratory Medicine, and former Chair of the Division of Clinical Microbiology at Mayo Clinic. And she happens to be the President of ASM and has about a list that goes on forever and would be boring to mention all on all credentials. So it's a very esteemed speaker, and most importantly, not to that, a great and nice person, which I really appreciate, having all that together. And the prize she's receiving today is the Hamao Umezawa Memorial Award, which is co-sponsored by the Japanese Institute of Antimicrobial Chemotherapy, and will be, actually we have a colleague from Japan, and they came extra as a whole team, who will be talking about this prize for a second.
Good morning, ladies and gentlemen. Thank you so much for giving me a time for presentation. I would like to introduce our institute and the accomplishment of Hamao Umezawa on this occasion. First, congratulations, the winner Robin Patel, for the historical Hamao Umezawa Memorial Award this year. Institute of Microbial Chemistry has been supporting Hamao Umezawa Memorial Award since 1989. The award is intended to honor individual researchers, scientists, or clinicians who have made outstanding contribution in the field of antimicrobial chemotherapy. Dr. Umezawa's spirit is reflected in Professor Patel's outstanding work. I hope that Dr. Patel's research will continue to develop. Dr. Patel, congratulations again.
Thank you very much. So with no further ado, go ahead.
Thank you so much. It's truly an honor to receive this award. I feel that this award is not just for me, however, but for everyone who I've worked with over my career, including all the students, residents, fellows, visiting scientists, and my collaborators, both at Mayo Clinic and outside of Mayo Clinic. So thank you so much. As I was reflecting on what to talk about today, I considered two options. One was to recount what we've done over the past 2.5 decades, and the second, which I chose, was to talk about a view for the future. So what I've decided to present today is a strategy, a potential strategy to help in the fight against antimicrobial resistance. And that's through better diagnostics and better use of diagnostics, a term I've titled microbial theranostics here. I'll make my case about why we need to do this and how we can do this, and hopefully convince you that this is a possible strategy to help us in this challenging situation. With this audience, I don't think I need to convince you that antimicrobial resistance is a problem that we need to work on. So I just picked two pieces of information. One, an infographic that comes from the United States Centers for Disease Control and Prevention on the left-hand side. And two, a statistic that's already been highlighted at this meeting from a very recent publication in The Lancet from the antimicrobial resistance collaborators. The infographic tells us what we know, but I think it's always important to align and make sure that we understand where we are. Antibiotic resistance is a global threat. It's happening right now. It's happening across the globe. We don't know the full impact of this. There's no system in place to track it. And unless we do something, our modern medicines, some of which were presented here today, will become obsolete and will really turn back the time on the treatment of infectious diseases. This is a growing crisis worldwide, so I think it's a problem worth solving. This is the right meeting, of course, to highlight this. The publication in The Lancet looks back at the year 2019, prior to the COVID-19 pandemic, and tells us that in that year alone, there were almost 5 million deaths associated with bacterial antimicrobial resistance. Those numbers have likely increased during the COVID-19 pandemic, and in any case, even without COVID-19, they were projected to continue to increase over the next several decades. So we have a challenge in front of us. And of course, development of new antimicrobial agents is key. But I'm going to propose that there's some other strategies that we can also take. Before getting into this, we need to really understand the enemy. And here, I think it's complicated. There are so many different types of bacteria, and we could put fungi and viruses here as well, that are associated with antimicrobial resistance. So many different clinical syndromes and so many different antibiotics. And here are two lists, one from the WHO, which is their priority pathogens list for R&D of new antimicrobial agents on the left-hand side, and on the right-hand side, CDC's urgent, serious, and concerning threats. And you'll note that they're not the same. There are some commonalities, but not all. And I would submit to you there are organisms that are also missing off of these lists. I can't possibly talk about all of these today. I will talk about a few that I've highlighted in red. But I think, this is a formidable challenge because we have so many enemies here to deal with. But the strategy that I'm going to present to you today, I think can be applied to all of these, whether I discuss them today or not, and even organisms that are not on this list. So as I think about antimicrobial resistance, I think about how we got to where we are today. And as a medical doctor, I feel responsible, and I'm going to take the blame on behalf of all medical doctors for some of the problem that we find ourselves in today. And part of this is the diagnostic paradigm that we use, that we teach our medical students and that we use in our practice today. We tend to spend time sorting out whether a patient's clinical presentation is caused by an infection, and then we generate a list of possible causative pathogens. And it's only in the later stage that we come to, how should I treat that patient? I will submit to you that this problem is contributing to antimicrobial resistance, this paradigm, but it's also failing because of antimicrobial resistance. We continue to have to use broader and broader antibiotics to treat because it takes us so long to get to that third step. Now, I'm going to use diagnostics as one potential strategy to combat this. We're not going to solve antibacterial resistance. We have to learn how to live with it. And that's diagnostics. It's estimated that 70% of clinical decisions are substantially based on the results of diagnostic tests. And yet, diagnostic tests only account for about 4% of healthcare costs. They are, however, rapidly increasing in number. And the cost of diagnostics is rising, an estimated 15 to 25% a year, faster than other areas of medicine, and mostly this is due to molecular tests, which I would submit to you, and I'm going to explain, is probably a good thing. Now, people talk about antimicrobial stewardship, and then they talk about diagnostic stewardship, and when we think about diagnostic stewardship, we think about people over-ordering diagnostics, and that does happen. It's estimated that 21% of laboratory tests are unnecessary. But there's another statistic that I think is important, and that is underutilization of what we have today. It's estimated, and this number I think could vary depending on how you generate it, but that 45% of laboratory tests are underutilized. And if these are truly the tool we think they are to help in infectious diseases management and also antimicrobial resistance, that's an important statistic. And what I'm going to highlight is how we can maybe use what we have, but develop new diagnostic tests to help us with antimicrobial resistance. So back to this diagnostic paradigm. So as a physician, we do a history and physical, we might look at some imaging and perhaps some preliminary laboratory tests, and then we generate our lists of which organisms could be causing this, and we order tests, cultures, serologies, molecular diagnostics. And then finally, once we figure out what it is, then we think about, well, how do I treat that? And we may need to wait on culture and susceptibility results, and all of this takes a lot of time. So I would submit that we can leverage new diagnostics to really do better. And by the end of this presentation, I hope to convince you that we can take out that third stage, and honestly, maybe that second stage as well, to get right to the answer. Now, fortunately, despite the crisis in antimicrobial resistance, we find ourselves in a great time because I'm going to propose creative use of technology can help us with this, and we are in the middle of a technology revolution. So I first want to explain what I mean by that. I've been practicing medical microbiology or clinical microbiology since the 1990s, and it's really an amazing field to be in because we've seen so many changes. Many of these changes are not from our own field, but we're adopting them to really do better in clinical practice. We've seen a revolution in proteomics, in nucleic acid amplification tests, and sequencing-based diagnostics, in diagnostic tests based on imaging of single cells and metabolomic analyses, advanced host response assessments for infectious diseases, and point-of-care diagnostics as well. And I'm going to briefly highlight these really as a way of saying technology revolution is here now, and we need to leverage this to do better to help us with antimicrobial resistance. So in the proteomics domain, it's really MALDI-TOF, mass spectrometry, that has changed how we do things in the laboratory. If you had asked me, or quite frankly, I think most clinical microbiologists in the year, say 2005, whether we'd be routinely using proteomics to identify bacteria and fungi in clinical laboratories, at least I, and I think a lot of my colleagues would have said, no, I don't think that's the case. But this technology, which really did not come out of microbiology, has completely changed our field. The large-scale description of use of this technology in clinical practice was published in 2009. Our lab and others, I'm sure Andreas's, began using this around 2010, 2011 in routine clinical practice. And in the United States, by 2013, this was cleared by the United States Food and Drug Administration, and now is a standard technique throughout the world. It's great, it's better, it's faster, and it's less expensive than anything that we had before. And we can look at a colony growing on a plate and in 5 minutes know what that is, actually in a better way than we could ever do before. So this is fantastic. We've also seen huge advances in nucleic acid amplification tests and sequencing-based technologies. So PCR was described in 1983, and that was huge for microbial diagnostics. In our lab, we began routinely doing PCR for infectious disease diagnosis just around 1990. And of course, with real-time PCR, we switched over to that. And sequencing also has had some amazing advances. So Sanger sequencing was described in the 1970s. That wasn't practical for clinical microbiology use, but with the advent of capillary sequencers, it became possible to perform sequencing in clinical microbiology. And we've been routinely doing that since the 1990s. That might not sound like much, but the human genome wasn't sequenced until the year 2001. And now today we have massively parallel sequencing, or next generation sequencing, if you will, that's being adopted into clinical practice. And so we have just a huge number of advances coming at us. This is a list of the real-time PCR assays that we developed and offer in our clinical practice at Mayo Clinic in the first 2 decades of the millennium. And you can see that there are organisms for which we absolutely need molecular diagnostics. We have nothing else. But there are also ordinary bacteria that we could otherwise culture, but by targeting molecularly, we can get a faster answer, and in some cases, a better answer as well. And so I'm going to argue that we can use some of this to really get ourselves to a better place as far as antimicrobial resistance. Now today, we don't have to perform nucleic acid amplification testing in sophisticated laboratories. Thanks to companies, many of whom are here today, we have very easy to use, quality controlled systems that can be deployed almost anywhere. Many of them are very quick, so they provide rapid answers, and some of them are highly multiplex, so we can answer lots of questions at the same time, which is often relevant to that differential diagnosis that we were generating in that classic diagnostic paradigm. And again, we've seen massive improvements in DNA sequencing as well, and this is being leveraged in the diagnosis of infectious diseases to really do a lot better in terms of finding out what's going on with our patient, and most importantly, finding out how to treat them properly and use our antibiotics very wisely. So just an example of this is the 16S ribosomal RNA gene present in all bacteria, which provides a nice target for identification of bacteria, but also for detection of bacteria in clinical samples. We've been routinely sequencing this since the 1990s from cultured isolates, but over the past 5 years, we've been routinely performing a PCR for 16S ribosomal RNA gene and then sequencing it in our routine clinical practice to determine which bacteria are causing infections, especially in culture-negative cases. We began using a Sanger sequencing-based approach that worked well, and we published our experience with this in Clinical Infectious Diseases last year. But we did note that there were cases where we had mixed sequences with Sanger sequencing, and so we turned to the addition of next-generation sequencing. We do a PCR upfront, so if the signal, the CT value, is very strong, we go straight to Sanger sequencing, but if it's weaker, we go to next generation sequencing, and if there's no signal there, we call that negative directly. And we have our work with this in PRESS in clinical infectious diseases. By adding next generation sequencing to Sanger sequencing, we were able to increase our yield, our rate of detection, by 87% in over 2,000 normally sterile body fluids and body tissues. This is a lot of data, a lot of information. This field is rapidly advancing. I think, back to diagnostic stewardship, when we have these new diagnostics, we do have to determine how to use them in clinical practice. So our group really has emphasized the use of diagnostic algorithms, and this is one example. This is for the diagnosis of infectious endocarditis. And of course, we start with blood cultures, but if blood cultures are negative and serologies for Coxiella burnetii and Bartonella are negative, we're often left with that entity of blood culture negative endocarditis. And if a heart valve is being removed, of course, it should be examined histopathologically, but our work and the work of others has shown that at that point, a sequencing-based diagnostic looking at the 16S ribosomal RNA gene is really the test of choice to go to next. So I think that these new technologies are informing, what's going on so that we can better use our antibiotics. Another type of sequencing, which is, I think, very attractive to a lot of infectious disease doctors, is shotgun metagenomic sequencing. With this technology, we take a clinical sample, and instead of targeting a single gene, we sequence everything in it. And when we do that, we sequence human DNA, we sequence whatever microbial DNA is present, whether it's bacterial, parasitic, viral, or fungal. And if you start with RNA, of course, you can detect RNA viruses as well. This is really the truly agnostic diagnostic, and I would say we're learning when and where to use this in our clinical practice, but I'm going to come back to this later because it does provide some unique possibilities in terms of antimicrobial resistance detection. It is, however, more complicated. It's more complicated from the wet lab perspective as well as from the bioinformatics perspective. So this is an infographic in another article we have in PRESS in Clinical Infectious Diseases comparing shotgun metagenomic sequencing and a targeted sequencing approach on the left and right, respectively. And again, more complicated, and we need to sort out when to use both of these, either of them, and when to use one over the other in our clinical practice. Some other examples of technology revolution have to do with single cell imaging. It's hard to imagine that we would be watching single bacterial cells grow with and without antibiotics to quickly determine antibiotic susceptibility. This is a platform from Accelerate called the Accelerate Pheno that's designed to test positive blood culture bottles And what it does is it puts aliquots of the positive blood culture bottle specimen into various wells and watches the individual wells. And what you can see is just one example of E. coli with 4 micrograms per ml of piperacillin tazobactam, is if the organism is resistant to that concentration, it grows up into a colony, as shown on the bottom video. But if it's susceptible, it doesn't grow, and in this case, it forms these long filamentous characteristics. And that's interpreted by a computer. I think that's the other thing, is that technology advances in terms of informatics are really making a huge difference. So this isn't something that my team interprets manually. This is all interpreted by a computer to get us to an interpretation, susceptible, resistant, and then MICs as well. And likewise, there's metabolomics. So we're all familiar with the odors of certain bacteria. Bacteria give off volatiles, and those can be sensed as an indicator of identification or growth. And this has been configured into another susceptibility platform from Specific Technologies, now acquired by bioMérieux, that takes organisms and antibiotic concentrations in a microtiter well plate format, as shown on the right-hand side, but puts little sensors above each of the wells in the plate to sense microbial growth as an indicator of susceptibility or resistance. This is not cleared by the United States FDA, but it is available in some countries, including in Europe. And then there are point-of-care diagnostics. I think with COVID-19, we saw tremendous development in these areas. But this is actually a publication that came out of a colloquium that the American Academy of Microbiology convened in 2016, looking at the revolution going on in point-of-care diagnostics. And of course, point-of-care diagnostics are not new with COVID-19. I mean, we've been doing home pregnancy tests forever, and home HIV testing has been out there. But we're going to see lots of changes here as well, and this is due to technology advances that allow us to deploy testing at the point of care. And I think this will make a huge difference going forward. My colleague, Dr. Brad Karen, and I wrote a commentary on this that was published in 2017 in the Journal of Clinical Microbiology, heralding this as really a major advance in diagnostics. And then we're also seeing advances in assessment of host response. So we're all used to measuring CRP or procalcitonin, but we can really look at unprecedented ways at host response, how the human is responding to whatever is going on. And although it's early days, it's thought that this kind of analysis will be able to point us to viral, bacterial, fungal, parasitic infections and non-infectious processes, and possibly, we don't know this for sure yet, but possibly point to specific microbial etiologies. I think that if this does pan out, it will have to be done on a syndrome by syndrome basis, based on the site of presentation and the specific specimen that's being analyzed. But I think it's a pretty exciting area of diagnostics as well. Now, this is really a basic publication that we published this year in Scientific Reports. And here we were looking at failed joint replacements, so hip and knee replacements, and whether we could use human proteomic analysis to separate out those two entities. And we just use a very small protein panel from Olink Proteomics, not designed for this purpose, but we could do this very, very well, and we could find certain proteins, such as CCL20 and oncostatin M and interleukin 6 and 8 and ENRAGE, that differentiated infection from non-infectious failure of these implants. And then we could pick out a small number of these proteins, again, from a small panel, so very preliminary, and get a very good diagnostic test with an AUC of 0.988. This is preliminary. It's not something that exists today, but it's just showing you that I think we can really look at host response, be it proteomic or transcriptomic, to try to ascertain what's going on from a microbiology standpoint. And I'm hopeful that we'll also be able to determine the cause of infection. So this is a human transcriptomic study that we have in press in the Journal of Bone and Joint Surgery that's suggesting that we can differentiate infections of joint replacements caused by Staphylococcus aureus versus Staphylococcus epidermidis. Again, very early on, but I think we're going to see big advances in this area as well. All right, so hopefully I've convinced you that we are in the midst of a technology revolution. So what can we do with this to help us with the crisis in antimicrobial resistance? Well, I think we can use these tests to inform the better use of antibiotics. And this will avoid unneeded treatment of patients who don't have a bacterial infection, or patients who have a bacterial infection that's resistant to the antibiotic that is being selected for choice, or downgrading to an older antibiotic and repurposing some of our older drugs. This will also likely decrease the transmission of infectious diseases, which has a societal benefit, and I think lead to new discoveries as well, which has future benefits. But sometimes when we look at our diagnostic tests that are coming to us, we're not sure how they will really make a difference. One example of this, if I go back about a decade, is the multiplex PCR panels for testing positive blood culture bottles. In the United States, the BioFire panel was cleared by the FDA in 2013, and when that came out, we weren't sure if we should adopt this in our clinical practice or not. We weren't sure if it would make a difference, but we thought it was really great technology. And so what we did to answer that question is to execute a randomized controlled trial. And we randomized patients to have this new diagnostic or not have this new diagnostic. You'll see there are 3 arms here. So in the 3rd arm, they actually got this test and real-time antimicrobial stewardship, because we weren't sure whether the test alone would be enough to get clinicians to change their practice in terms of prescribing antibiotics. And the thing about this test is it doesn't give you any different results than you're going to ultimately get. It just gives you faster results. So the question is really, what is the value of that speed? And it's an expensive platform. What we found is that there was no difference in mortality or length of stay or admission to the ICU, things we would have loved to have seen as outcomes. But there was a difference in terms of use of antibiotics. So this is a profile of the 3 groups, the control, the rapid test, and the rapid test plus stewardship in each row here. And what you can see in red is the time to identification of what was present in the positive blood culture bottle. It was a lot faster, obviously, with the rapid test. In the triangle with the E inside of it, that's time to antibiotic escalation. That was much faster with the rapid test, with or without antimicrobial stewardship. But the time to de-escalation, which is shown in the upside down triangle with a D, was only faster with the rapid test and the combination of real-time antimicrobial stewardship. So we learned a lot, and we adopted this in our clinical practice. But this panel at the time only had 1 gram-negative resistance marker, KPC, and we didn't have any KPC-producing organisms in our study. This was 613 positive blood cultures in this study. So the benefits were more on the gram-positive than the gram-negative side. I think, and I was talking to some people at this meeting about this as well, it's important to think about how these results are reported from the laboratory. What we get off of the machine is shown in the left-hand column, but what we actually report is shown in the middle and right-hand columns. And as we're seeing more and more of these tests, what's coming off of the machines is very complex, and clinicians oftentimes don't understand that. They don't speak antimicrobial resistance gene languages. And so we need to translate what's coming off of the instrument into something that they can understand. And for example, if we detect Staphylococcus aureus, and MecA-C MREJ, we report that out as Staphylococcus aureus. We say that MecA is detected. We say that's MRSA. We tell them what it means that it's MRSA, and we tell them also how to treat that bacteremia. And there's more information on this that is in press in an article that the Antibacterial Resistance Leadership Group has in press in Clinical Infectious Diseases, because it's getting very complex to report around some of these tests. And making sure you're reporting correctly is really important so clinicians understand what's going on. We also did another randomized controlled trial. This was looking at the Accelerate Pheno platform, that real-time single-cell imaging platform that I showed you earlier with the videos. But we did this in patients with gram-negative bacteremia, so blood culture bottles positive for gram-negative bacteria, because the original version of the BCID didn't really help us much with gram-negative bacteremia. So here we randomized patients to have the Accelerate Pheno test or not. They had to have gram-negative bacteria in their positive blood culture bottles. And what you see in blue is what happened with this new test. The time to antibiotic change was faster, and the time to gram-negative antibiotic change, not surprisingly, this was a gram-negative study, was markedly faster. The time to antibiotic escalation shown on the left-hand side was faster with resistant organisms, and the time to antibiotic de-escalation was faster with susceptible organisms. So this kind of study, a randomized controlled trial, can really show the benefit of diagnostic or the lack thereof, and also provide guidance on how to use these diagnostics. But now I'm going to turn to where I think we need to go with diagnostics. I've shown you that we've gone through a technology revolution that continues, that we have ways of assessing value of diagnostics and how to use them in clinical practice to idealize antibiotic use. But getting back to that diagnostic paradigm, I think we can get to a place where we get the answer as to what's present and how to treat it all in one step. And we have some assays out there that already do this. For example, the Cepheid MRSA Staph aureus skin and soft tissue infection assay, and they have a nasal screening assay that does the same, tells you if you have Staph aureus and if it's MRSA. That's been around for a while. You're likely familiar with their TB assay that tells you if you have TB and rifampin resistance right away. I think this kind of assay is really helpful facing antimicrobial resistance. There are also assays out there to detect, for example, Neisseria gonorrhoeae and ciprofloxacin resistance or susceptibility, and Mycoplasma genitalium and macrolide resistance or susceptibility. I think we can probably do this with most microorganisms. And let me just show you a little bit more data on this. This is an example of an assay that we developed to look at Mycoplasma pneumoniae, detect it, and call out the most common mutations associated with macrolide resistance. And using this assay, we showed that in our practice, 10% of our Mycoplasma pneumoniae detections were predicted to be macrolide resistant, and this was looking at specimens collected between 2014 and 2021. So that, I think, is a great way, not just diagnose, but tell people how to treat or how not to treat based on detection of resistance. And now I'm going to turn to another organism where we've done some work in this regard, and I was happy to hear about new antibiotics for Helicobacter pylori, because I think after you see this, you'll be convinced that we likely need that. So, and it's great to talk about H. pylori, of course, in Australia as well, where this organism was really recognized as being the significant pathogen that it is. It causes peptic ulcer disease, non-ulcer dyspepsia, gastric carcinoma, and gastric maltoma. And it is on the WHO priority pathogens list, but not on the CDC's list, interestingly. But it's also called out as a group 1 carcinogen by the WHO. So it's a drug-resistant carcinogen, which I think is pretty dramatic. Now, it's typically treated with combinations of 2 to 3 antibiotics with a proton pump inhibitor. We know that 20%, it's probably more, fail an initial attempt at treatment. And we know, obviously, that factors associated with treatment failure are patient compliance and resistance to the antibiotics that are being selected. But we have a problem. Resistance in Helicobacter pylori is not tracked. It's oftentimes not even ascertained. So as a reference lab, we do a lot of testing of Helicobacter pylori isolates for antibiotic susceptibility. This is biased data because you can only get an isolate if you get a stomach sample and then do a culture and recover the organism. And the preferred way to diagnose Helicobacter pylori infections is with non-invasive diagnostics, so not everybody's getting a stomach biopsy. But this data was very alarming to us. We looked at 413 Helicobacter pylori isolates. These were recovered between 2011 and 2016. And what you can see here is that the rate of clarithromycin resistance is over 70%. And the rate of metronidazole resistance is extremely high as well. Ciprofloxacin we don't have a breakpoint for, but over 50% of the isolates had high MICs above 2 micrograms per ml. So this is very alarming, but at the same time, it's really biased because it's just isolates of Helicobacter pylori. But looking at this, we felt, well, we're looking at what's happening downstream, the failures of antibiotic therapy to treat these patients. And what we really need is a diagnostic that can call out resistance or susceptibility at initial diagnosis, just like I was making the case for these other organisms. So we set out to design an assay that would detect Helicobacter pylori in stool, providing a non-invasive diagnostic, but also call out clarithromycin resistance at the same time. And fortunately, there are a limited number of SNPs that are associated with this, so we were able to design a PCR assay that could do just that. And the vision was that this would fall then under the list of non-invasive tests, such as stool antigen testing and urea breath testing. We could have a stool PCR that would detect Helicobacter pylori, but also call out clarithromycin susceptibility or resistance. Of course, we can always get to this by doing a gastric biopsy, but most patients don't undergo a gastric biopsy to get to that point. So what did we find? Well, first of all, we had the question of how common is clarithromycin resistance in Helicobacter pylori at initial diagnosis. I don't think we've really known the answer to that question very well. And then we also had the question of what is the implication of clarithromycin resistance in Helicobacter pylori. So we looked at 223 stool samples from 197 patients at our institution, and then we looked at the impact of finding predicted clarithromycin resistance in their Helicobacter pylori. This was predominantly adult patients, a mix of females and males, and what we found, first of all, is that clarithromycin triple therapy regimens were the most frequently used initial therapy for Helicobacter pylori, so this is relevant. This is what people are using today to treat Helicobacter pylori. How much clarithromycin resistance did we find? Well, overall in the study, it was 37%, but in the initial diagnosis group, it was 31%. So pretty significant considering how commonly people are selecting to use clarithromycin-based regimens. And then we looked at treatment outcomes. So what happened to patients in whom clarithromycin resistance was predicted? And in the group overall, the success rate was 48% when there was predicted clarithromycin resistance versus 74% when no resistance was predicted. And in the group receiving clarithromycin-based triple therapy, the success rate was 41% when there was predicted resistance and 70% when there was no predicted resistance. And as you might imagine, since we do test of eradication testing, when we looked at failures, the rate of clarithromycin resistance was higher there. It was 64% in the group as a whole and 69% in those who had been previously treated with clarithromycin-based triple therapy. I think this is just the beginning. I think we can design tests that can give us the answer of what the organism is susceptible to or resistant to right at the beginning. And we can also do this with some of the advanced molecular tests that we have. This is an example of one patient out of 408 from a large shotgun metagenomic sequencing study that we did in periprosthetic joint infection. This gentleman had a right knee periprosthetic joint infection. It was obvious he was infected. He had swelling and pain and sinus tracts, but he had been aspirated multiple times, and though he had elevated cell counts, all of his cultures had been negative, including those done at our institution. So he underwent a 2-stage resection arthroplasty and ultimately ended up failing that. But we received specimens from that procedure, and we executed a shotgun metagenomic sequencing analysis on that and showed that he was infected with Metamycoplasma salivarium. Well, we didn't know for sure if that was the pathogen, but later it was recovered in culture and proven to be the pathogen. And what I want to show you is another analysis you can do on shotgun metagenomic sequencing data, because when we had that information, our clinicians asked us if we could look at the sequencing data and tell them whether his isolate was likely to be macrolide susceptible or not. Of course, we don't know a lot about macrolide resistance in Metamycoplasma salivarium, so we turned to Mycoplasma pneumoniae, and we looked at the mutations in the 23S ribosomal RNA gene associated with macrolide resistance there. And we predicted, based on what we saw, that he should have a macrolide-resistant organism, which he did when we ultimately recovered it on culture. So one advantage of some of these deep sequencing approaches is that they can also give you information on susceptibility or resistance. And this field is advancing slowly but surely. This is some data looking at Staphylococcus aureus, whole genome sequencing data, and the ability to take that data and predict susceptibility or resistance to a traditional panel of antibiotics shown on the very left-hand column, which now we're able to do. And increasingly, as we understand mechanisms of resistance, we can get to that point. And of course, we're seeing advances on the sequencing side. So the Oxford Nanopore Technologies MinION can provide a rapid assessment of sequence. And this is a study we did looking at bile from patients undergoing pancreatic head resection. And within 6 hours of collection of the specimen, we could recapitulate enough data to enable clinicians to pick out the same antibiotics that they would based on culture and susceptibility data. So preliminary, but we're getting there. So finally, I wanted to comment on discovery through testing. There's a lot in infectious diseases that we still don't know. We have a lot to discover. I think this is another piece that makes what we do exciting. And so our group has discovered several new organisms: Borrelia mayonii, Ehrlichia murus subspecies oclarensis, Yersinia rochesterensis, and I know that we have many other novel species that are clinically relevant that remain to be described. We also, day by day, are understanding antimicrobial resistance better. We have described a new vancomycin resistance element in Paeonibacillus papiliae. Through our PCR diagnostic testing, we were able to recognize a new disease, the etiology for a new disease, hyperammonemia in lung transplant patients and its association with ureaplasma species. And then we're also able to recognize things like seasonality of infectious diseases, which perhaps some of you know, but I didn't actually know, and I found very interesting, so I thought I'd just share this with you here. We do a lot of testing for Bordetella pertussis and also for Legionella by PCR, and we monitor positivity rates for quality control purposes, which is very boring for most people. But we see clear seasonal trends. Of course, the Bordetella pertussis data is prior to the COVID-19 pandemic because that completely disrupted the epidemiology of that organism. But what we see is in the United States, peaks in the prevalence of positivity for both of these organisms in the summertime, which is, I think, really interesting, and we're able to make other observations that we maybe didn't fully understand before. And then finally, we're learning. I mentioned that we need to reconsider our diagnostic paradigm. Sometimes we're also wrong as medical doctors, and this is an example of that situation. This is a 59-year-old man from Minnesota, where I live, which is in the central northern United States. And he had had a rash on his face and a little bit on his arms as well for about a year, and some neuropathy. He lived in Minnesota. He had traveled only to Mexico. A biopsy was done that showed granulomas, and there were many acid-fast bacilli present. And so he was diagnosed with leprosy, and I can tell you that leprosy is an unusual diagnosis for us in Minnesota. But that was his diagnosis. And just more out of curiosity than anything, our clinicians asked us if we could do our 16S ribosomal RNA gene PCR sequencing assay on his skin biopsy, which we did. And of course, we would have expected to find Mycobacterium leprae. But we did not find Mycobacterium leprae. This was Mycobacterium lepromatosis. And so it just is an example that shows us that sometimes in medicine we know the answer because, I don't know, we're medical doctors and that's what we were trained to do, make a diagnosis, but we can be wrong. And then we can discover new findings as well. This is a brain biopsy specimen that I was reading out. It's coming from, again, our 16S ribosomal RNA gene PCR sequencing assay. And I'm showing you kind of raw data here to show you how complex it can be. On the top is the forward reads, and on the bottom is the reversed reads that we got. And you can see that the most abundant organism is something that is called Candidatus amebophilus species, which is an endosymbiont of Balamuthia mandrilaris. And this is really interesting. So this patient is a transplant patient, a lung transplant patient who had come in with seizures and had these findings and had undergone brain biopsy, which was non-diagnostic. And so then they were really grasping at straws, looking for a diagnosis, and had executed this sequencing-based diagnostic. Of course, you know, we don't think this was an infection per se with this bacterium, but instead a parasitic infection. And so we did a Balamuthia mandrilaris PCR, which was positive. And we also re-reviewed the histopathology and were able to see the organism that is right in the middle of that histopathology slide here, but had been missed on initial diagnosis. So we're also discovering new entities or making better diagnoses on our patients through better diagnostics. So let me come back to the scenario that I proposed in the first place. Again, this is our classic diagnostic paradigm. Is the patient's clinical presentation caused by an infection? What are the possible organisms then that are causing that infection? And then how do I treat whatever it is that's causing that infection? I think that with advanced and better diagnostics, we can get rid of that third step. We can move it to the second step. And I think arguably we could actually collapse all of it to one, where we get the answer and we know how to treat patients right away. And with antibiotics, I think this can be really helpful because there are many patients receiving antibiotics that they don't need. And there are some patients not receiving antibiotics that they do need. And this type of strategy would really give us the answer up front. I showed you some examples of how this might be done. I showed you our work with Helicobacter pylori, for example, showing that, we're mistreating a lot of patients with Helicobacter pylori because, quite frankly, we don't even know what kind of resistance they're carrying with their isolates. But we now have technologies that can get us these answers, and I think we need to think about leveraging them to get us to that point. It doesn't mean that we use every diagnostic that comes our way. We have to show value, as I showed you in randomized controlled clinical trials, if it's not obvious that a test will be valuable. But I think we have a lot of leeway, a lot we can do with better diagnostics to help us combat antimicrobial resistance. And with that, I'd like to thank the NIH for supporting my work, and my two laboratory teams, my research laboratory team and the clinical laboratory team as well. Thank you very much.